presentation. Now we continue with our fourth presentation. It's Simon Ströbel. He will give this presentation. He is a senior scientist and product manager at InSphero AG. He is working there on 3D model and assay development for tumor and skin microtissues. He has a master in cell biology and did his PhD in biomedical engineering at University in Basel, where he was working on 3D perfusion bioreactor systems. And I'm curious to hear what you say.
Thank you very much. So it's working? Yeah. So thank you very much, Max, for the very kind introduction, and also thanks to the organizer committee to give me the opportunity to share some of our results today. I'm going to talk about stem cell-based microtissue engineering for drug development, and in particular looking at iPS-derived cardiomyocytes for drug de-risking. So why are stem cells an attractive source for tissue engineering? So of course, primary cells are limited in availability and supply, especially for if you look at heart or neurons. They're genetically more consistent, but also enables genetic modification, which basically then reduce polymorphism, but also enables disease modeling by specific gene targeting. But it also enables a scalable production of tissues, where you can then implement these tissues for early or also late drug development. We know different types of stem cells, of course, looking at embryonic stem cells over stem cells which are tissue resident, like in hair follicle or intestinal crypt cells. But also induced iPS cells are more attractive to use as model systems. So when we talk about stem cells and tissues, 3D is always the requirement, and so the integration of such stem cells in 3D microtissues will enable to have a more similar situation to the native situation. It also enables the use of translational biomarker, so translate in vitro into in vivo. And also important, 3D environment also is a more physiological state, which enables to have more relevant assay setups. And also, of course, generate in vitro model with the highest clinical relevance. And this will lead to novel drug testing concepts. So to just give you an example how to use stem cell-based tissue engineering for drug safety testing, I'd like to introduce the iPS-derived human cardiac microtissues. We at InSphero, we developed a technology to produce microtissues at a scalable and throughput-compatible way. This technology is based on the hanging drop, a well-known hanging drop method, where cells basically reaggregate in suspension. So we use this concept or this method to re-engineer cardiac microtissues by using iPS-derived cardiomyocytes together with cardiac fibroblasts. So here you can see a transversal section through such a sphere. We have positive or myosin positive cardiomyocytes and also fibroblasts together. We then also went on to test the functionality of this tissue, so looking at the beating rate, and we realized that having a multicell type microtissue, which means cardiomyocytes plus fibroblasts, represents a more physiological beating rate than if you only culture iPS cells themselves. So this also is shown here, where we have like a visual assessment recorded over a longer period, and you see that this tissue show a beating rate of around 60 beats per minute, and we used this and exposed the tissues to Doxorubicin, which is a known drug which induces heart dysfunction, and this led to an increase of the beating rate initially, but also induced heart arrhythmia or arrhythmic pattern of beating after a longer incubation time. Another functional assessment is to look at measuring the actual potential. And so this is data from a collaboration with Clyde Bioscience. They developed a cell-optic device, which is a microscopic device, assessing fluorescent signal in the microtissues. And so this fluorescent signal, which is based on a voltage-sensitive dye, shows signals related to the depolarization of the cells or the tissue and the repolarization. So everything which is in the green fluorescent boundary represents the depolarization. Everything which is at higher wavelengths represents the repolarization. So if you integrate the signals over time, you see the depolarization, but also repolarization pattern. To now have this calculating the action potential duration, you require to make the ratio of the short, which means the green signal versus the red. This is to minimize moving effects from the tissue itself. So we used this technology and compared, first of all, this action potential duration from 3D microtissue to 2D, and we could show that this is shortened in microtissues. We subsequently also applied a calcium channel blocker, nifedipine, and could show that only in 3D microtissue we could show a relevant effect, which shows basically a higher sensitivity in this 3D model. Similarly, we also applied potassium channel blockers and could show the opposite, that we have an increase of the actual potential duration in our 3D microtissues versus the 2D situation, and this will result in a lower sensitivity. So we've seen now cardiac microtissues, but of course we at InSphero also provide various other microtissue based on primary cells, but also as you can see here for the brain microtissues, iPS based. So looking at microtissues to use in the development of the drug, they can be used in actually every step of the development of the drug, starting from target discovery, using screening approaches, but also in lead optimization or drug safety, but ultimately also use the models for investigative toxicology. So finally, I'd like to acknowledge Irina Agrokova from InSphero. She did most of the model engineering, also Jens Kelm for supervision. He cannot be here, unfortunately, today. Also thanks to Godfrey Smith from Glide Bioscience for providing the data. So thank you for your attention.